Happy Monday. Okay, we are just going to do this here because I had something that I couldn't get out of that I needed to present for some research that I do. So we're just going to let you sleep in and watch this video instead of coming into class. We have officially moved from the Mendelian portion of the show to the molecular portion of the show. So from now on, we're going to be talking about DNA, transformation, CRISPR, things like that. The next 2-3 chapters should be mostly review for you. You know, like DNA is the transforming principle. Essentially, we are just talking about genetic information. We're talking about how we got here, how we determined the DNA was the genetic material, and then a little bit about how we use it. So, genes contain information that is passed on to the next generation. We knew this, and in the 1910 probably we started looking at this, like what is the actual genetic information? How is it passed to the next generation? 1940s chemical component of chromosomes is still unclear, but 1953 Watson and Crick put forth the hypothesis for the double helix of DNA with the research that they stole from Rosalind Franklin. Okay, so we knew four characteristics. Four characteristics. Okay, replication has to be part of the characteristics of the genetic material. That's what they knew going in. It has to be able to replicate. It has to be able to store information. It has to be able to express, right? It has to, like, the information has to go into something. So now we know DNA goes to RNA, RNA goes to amino acids, which make proteins, and proteins that's what expresses. That the protein of your eye color, your hair color, all those things. That is protein, and that's the expression. And it has to allow variation. So now we know how DNA replicates, and we'll talk about the variation in a couple chapters and how we actually get changes in DNA, which is the driver for evolution. Okay, central dogma of biology. Central dogma of molecular genetics sounds super complicated, so you're going to want to make this harder than it is. It's not that hard. It's what you already know. DNA goes to RNA, RNA makes amino acid sequences, those amino acid sequences fold into proteins, and that's how you go from the genetic information of DNA into your hair color, your eye color, diseases, so on and so forth. That is the central dogma of molecular genetics. Do not make it harder than that. It is not harder than that. That is just the main concept that we're dealing with here. So you have DNA that transcribes, transcription. That's the process from DNA to RNA. If I ask you to transcribe the notes for this class, you would do that in English because I'm saying transcribe. So that's DNA to RNA. It's essentially the same language, with or without a U. Same language, transcription. If I ask you to translate my notes, that would be in a different language. That is RNA to amino acids / proteins. Okay, that's how I keep it clear in my mind. Transcription is the same language, DNA to RNA. Translation is a different language, RNA to amino acids, and then the folding of the proteins. And this is the big picture of the central dogma. DNA via transcription goes mostly into rRNA, right? In the ribosome. In the ribosome, you actually get the rRNA, the tRNA, and the mRNA into a sequence of amino acids. Through the process of translation, those amino acids fold into proteins, right? This is the central dogma. You already know this information. Don't make it harder. Don't panic. Okay, genetic material is the source of variation. They knew that. They knew that it had to mutate somehow. We know that mutation in DNA reflects, that's reflected in transcription and translated into specific proteins. So if you have the wrong code of DNA, right, you have a C instead of a G or an A instead of a T, what that will do is we will do a whole chapter on mutations, but what it can do for things that we know, like sickle cell anemia, is it shifts it into a different amino acid. That amino acid doesn't fold correctly, and it doesn't make the correct protein. Do you see how that mutation works? And we'll, again, talk about that a lot more, but that's just what we're doing. Mutation is present in the gametes. They're present in your eggs and sperm. Those are your gametes through meiosis, and they get carried to future generations. Now, this genetic variation is actually the driver for evolution. This is the driver for genetic biodiversity, or any kind of diversity, really. So the question is it a DNA or is it a protein? The genetic material is physically transmitted, egg and sperm, from parent to offspring. This, again, okay, so they knew that. Now, it's like, is it nucleic acids? Is it protein? Almost everyone was on the side of protein here. Everyone was like, it's got to be protein. Nucleic acids, there's 4, A, T, C, G. The entire genetic information cannot be just A, T, C, G. That's crazy. No one believed it. So again, most everybody favored proteins. We have Meischer in 1868 that actually isolated the DNA in the nuclei and realized that was present in chromosomes and thought there's no way. Again, it's only 4 letters. There's just no chemical diversity here. There's no way this is the genetic information. Levine proposed a tetranucleotide hypothesis, saying that DNA contains equal amounts of the 4 nucleotides. Turns out that's not true. Chargaff actually will show that that is, in fact, not true. But he postulated this identical group of 4 components based on DNA structure, which turned out to be mostly correct. But again, the lack of chemical diversity in DNA suggested it could not store this kind of extensive information, and proteins were favored as the genetic material. Okay, now we are going to try and watch this video.
What component in our cells is the genetic material? Oh.
Although the answer seems obvious today, it's DNA. It took 3 scientists almost 20 years of hard work to discover this. The scientists, Oswald Avery, Colin MacLeod, and Macklin McCarty, began their search because of a surprising discovery made by Frederick Griffith in 1928. Griffith found that if a virulent strain of pneumococcus bacteria, known as the 3S strain, was heat-killed, mixed with live pneumococcus bacteria of the non-virulent 2R strain and injected into mice, some of the R bacteria were transformed into living infectious 3S bacteria which killed the mice. In addition, all descendants of the transformed bacteria were also infectious. Griffith knew that some component of the heat-killed S cells was responsible for this genetic change, but he did not know which one. He called the unknown agent the transforming principle. After Griffith published his findings, Avery and his colleagues set out to find the identity of the transforming principle. First, they broke open or lysed heat-killed 3S bacteria with a detergent, releasing the contents of the cells. Then they used a centrifuge to separate the cellular contents known as the cell extract from the cellular debris. To make sure that the extract contained the transforming principle, the extract was mixed with a culture of living 2R bacteria. Then the mixture was incubated and plated on a petri dish. Colonies of 3S bacteria appeared on the plate, while no 2R bacteria were able to grow, indicated that the 2R bacteria had been transformed. Avery and his colleagues now knew that one of the components of the extract, polysaccharides, proteins, DNA, or RNA, was responsible for the transformation. To determine which was responsible, they treated samples of the cell extract with various purified enzymes and crude enzyme preparations containing enzymes that could degrade one or more of the macromolecules. A sample of each treated extract was then mixed with 2R bacteria, incubated, and plated on a petri dish. 3S colonies appeared on the plate after all enzyme treatments except DNA-degrading enzyme. This result showed that DNA was the transforming principle. Let us consider a modern version of Avery's transformation experiment where we start with a mixture of DNA and RNA purified from a 3S extract. We know that either DNA or RNA must be the transforming principle. To test each nucleic acid independently, enzymes known as nucleases are used. First, RNase is used to break down the RNA in the extract. A sample of the RNA's treated extract is then tested. The presence of 3S colonies